Okay, so thank you so much. I'm very happy to discuss with you today, and I try to talk about brain-derived extracellular vesicle in the context of dementia and tauopathy. And I try to explain how we use different approaches to understand, but also for improve the diagnosis of these different diseases. So first, a very short introduction about dementia. What we talk about dementia, we talk about syndrome, which affect memory, behavior, and ability. But we don't talk about just Alzheimer's disease. We talk about different sort of dementia. Exists reversible dementia. For example, when we have a COVID, we observe different sort of dementia. But in general, this dementia is caused by degenerative disease. And exists different sort of dementia. Of course, Alzheimer's disease is a more common causes. And if we try to explain the biological cause of this degeneration of the brain, we can suggest a sort of idea that is very clear now, that is a question of disorder of homeostasis of the brain, because we have some protein very fragile in a brain. The protein is tau, the protein is APP, alpha-synuclein, or TDP-43. And we don't know why during aging, this balance between degradation and formation of the protein have some problem, and we have aggregation of this protein in the brain. This aggregation causes degeneration, but I think the important question that we can mention today is the first question is what sort of protein accumulate in the brain, and in fact is not completely clear, because during aging we can have different dysregulation of this protein. So of course you know very well that the APP or the amyloid and tau can accumulate in the context of diagnosis of Alzheimer's disease, and we talk today about that, about the diagnosis of Alzheimer's disease. But as you can see, we can classify the other sort of tauopathy also based on the fact that we have amyloid or tau. For example, in the frontotemporal dementia, we have also accumulation of tau in the brain. Another important question that I investigate for the research in Alzheimer and dementia is, of course, what sort of region, because the region affect in the brain and use different symptoms and different disorder in cognitive disorder of. For example, if you have a problem in a frontal cortex, you have problem of disinhibition, and this is not the case if your temporal cortex is affected. And the thing is, we believe that the protein accumulate mainly in neuron, but we know now it's not completely true, because this accumulation of protein exists everywhere in the brain. And in particular, we have also the more abundant cells in the brain, which are astrocytes or glial cells, who are also impacted in this pathology. A very important point also to mention today is the time and the progression of this pathology. It's a very, very low pathology, and some persons believe that the pathology started very long time before the first symptom of the pathology. And it's a very complex situation, because to diagnose early this pathology is very complex. So just a point, a brief introduction about what we have in 2023 about Alzheimer. So the diagnosis is still difficult, costly, and time-consuming, and we have no effective treatment or no disease-modifying treatment. But we can consider that the page or the story changed very recently, because the criteria of diagnostic change, and recently we can consider more the biological aspect of the biology that is different before. But also in US, we have also the first disease modifying treatment for the amyloid. That is also a lot of a long story, but we can imagine that is the beginning of the new story for Alzheimer's disease diagnosis and treatment. So just to introduce my group and my research, we are located in the Center of Psychiatric Neuroscience, which are a center where we have a lot of biologists and different bench and microscope, but we also work in close collaboration with the Center of Memory, so the providing the diagnostic of the patient, and our different question is to first understand the links between the protein imbalance and the cognitive disorder. But we all try also to repair the disturbance in the brain during aging. And today I try to talk mainly about the protein dysregulation diagnosis and how we can investigate this question. And for that, we have a small team in Lausanne, but we also collaborate a lot with a clinician. So how we can understand the links between this protein dysregulation and the cognitive and behavior disorder. So I try to introduce a very general method that we use. This method starts in humans, by MRI or by post-mortem samples that we obtain by different autopsy. And we try to find something in this brain of the patients, and we reproduce this model in animals. It's a very general method. And after, we try to understand if we have a link between this dysregulation of one region or one cell and the behavior of animals. And if we use this different technique, we publish different papers suggesting that, in fact, the astrocyte, so it's a cell very abundant in a brain, is also affected by this accumulation of tau protein. And very interestingly, if we reproduce this accumulation in animals, we can induce also different memory disorder in these animals. Another recent publication where we use a brain of patients with different sorts of tauopathies, so Pick's disease, PSP, or Alzheimer disease, and we isolate in this brain a sort of the vesicule, the extracellular vesicule, secreted by the brain. So in this brain, every cell can secrete different material, and in this different sort of material, we have extracellular vesicule. If we inject this vesicule in a brain of animals, you can reproduce the different pathology in the animal. And it's the first time that we reproduce the different pattern of expression of disease. And very interestingly also, if we use this vesicule and we inject in different sniffer cell, we can also observe a very different results for the response of this cell in front of this different vesicle. Okay, it's just a brief introduction about our research, but it's just to introduce the question of diagnosis. What is the interest of this research? Very fundamental for the diagnosis. So just to explain the diagnosis, and it's very important to confront the AD research and the AD diagnosis that is completely toward opposite, but very connected. Why I say that? I say that because in AD research, we have approximately 250,000 publications to explain that we can restore the pathology, that we understand the pathology in animals model. And all these publications focus on the development of different drugs. And as you can see here, you have the different topic of the drugs, the development, which are just the topic of the AD research since the century. And in the other side, you have the diagnosis. The diagnostic is a long and winding road where we have different steps, but it's a very long process because the pathology, so the first description of the pathology is in 1910, but the first criteria of diagnosis is in 1884. Where we describe for the first time the different sort of diagnosis only by autopsy and biopsy of the patients. So, of course, this diagnosis change, but take a lot of time, 27 years to have an update of this diagnosis in 2011, where we have at this stage a new description of the pathology. We talk of preclinical MCI and AD, so only 10 years ago. And very recently, we have also a new criteria for the diagnosis, and we talk about A for when you have amyloid, T when you have tau, and N when you have degeneration. So based on that, of course, we can classify the different patients, but it's always complex because sometimes you have patients where we have a symptom for the Alzheimer disease at the clinical level, but you have no all biomarker. So it's a process complex, it's a process long, and implicated different steps of the diagnosis. But the thing is, the research continue, of course, and since 10 years, we have observe a lot of publications who describe a new criteria of diagnosis. And during approximately 5 years, we observe a lot of publications who describe new letters as inflammation, neurodegeneration, vascular, alpha-synuclein, and a lot of publications propose to include these different later in the diagnosis. Very recently, during the last summer, different experts propose, is not completely validated, but propose that we come back just in 2 letters for the diagnosis of Alzheimer, which are the A and the T. But the big difference for the diagnosis would change is the fact that before we just consider the A and T in a CSF, but now we talk about fluid including, of course, the plasma. Because the research in a plasma we can find a lot of papers who propose that we can dose these different proteins, amyloid and tau, but other protein like neurofilament or neurogranuline directly in a plasma. And why? Because between this period, we have also observed a lot of new technology, and including the CMOA technology, which permit to detect with a high sensitivity the low quantity of different protein in biofluid. So just to continue my presentation, it's very important to consider that this measuring brain-derived protein in the blood is complicated for different reasons. The first one is the presence of phosphoto and APP in the plasma suggests that it had crossed the BBB, and we don't know exactly the mechanism that for this protein pass to the brain in the plasma. Another important point to consider that is this protein, the tau and APP, is also present in other organs. And in particular, the APP is very abundant in different organs. Also, the progression of the pathology is very different, and we talk about one region affect in the beginning of the pathology. So suggesting that perhaps diagnose the early stage of Alzheimer's disease is probably complex because we dilute the signal of this protein. Of course, the vascularization is also very complex because it's not homogeneous in the brain, and the plasma, it's a very complex biofluid, and it's perhaps this reason why for the cancer it's very difficult to find some biomarker. Altogether, this result suggests that the individual heterogeneity is very high for the plasma biomarker, but we can believe that we can increase this diagnosis in a plasma. So how to improve this diagnosis in dementia? So for that, I introduce a topic where we work since 5 years. It's the topic of extracellular vesicle, so brain-derived extracellular vesicle, because this small vesicle that I explained that we isolate in a brain of patient and that we can inject in animals can cross the barrier, and we can observe this vesicle in the different biofluid, including the plasma, the CSF, but also the urine and the saliva. And it's a long story, but it's already used for the cancer diagnosis, prediction, and also biomarker evaluation. So this vesicle is wonderful for different reasons. The first one is the vesicles, the extracellular vesicle, reflect the content of the cell which secreted it. So suggesting that we can have a sort of reflect, a mirror of the content of the cell. But the other thing that is at the surface of this vesicle, you can also identify the origin of this vesicle, and by a sort of antibody very specific for the different sort of vesicle, you can isolate the vesicle secreted by the neuron, by the astrocyte or the microglia at the different place of the brain, in theory. So in my group, we are interested by these different vesicles. So we have different models, the rodent culture, human iPS, human cell, and we try to isolate these different sort of vesicle and investigate the content of this vesicle. So just a brief result about Jeanne Potter, who work in PhD in my group, where and in collaboration with the hospital of Lille, where we isolate the different vesicles in different patients with different diagnoses, and we use mass spectrometry to analyze the content of this vesicle. So in the context of Alzheimer's disease, where we have different progression of the pathology, as you can see, we observe a progression of different biomarkers, in particular amyloid, increase in this vesicle during the progression of the pathology. The thing that is very interesting, that is, if you consider the different biomarkers present only in the early phase of the Alzheimer's disease, we find in this vesicle approximately 20 proteins present in vesicle who are only increased in the first phase of the pathology, suggesting that the BDEV contain numerous biomarkers for the progression of the pathology, but also for the prediction of the pathology. But how we can isolate this BDEV in a plasma? Because as I explained, in a plasma, we have also vesicle for the lung, prostate, liver. It's a sort of cocktail, a soup of vesicle from all organs. So in fact, if you want to isolate this vesicle, you collect the plasma of patients, you isolate the total number of vesicles, and after you use a technology to immunoprecipitate, so you use antibody against the different protein at the surface of this vesicle. And we have different candidates for the astrocytes, the neurons, the oligodendrocytes. And during approximately 5 years, a lot of paper proposed to isolate this vesicle and quantify the different protein in this vesicle. The main problem is 3 years ago, different papers suggest that this protein at the surface of the vesicle is not very specific to the brain. It's also secreted by the brain and stick in the vesicle. So we don't know exactly if this vesicle comes from the brain, and we have to increase our knowledge about this vesicle. So for that, we use different technology in my lab, and we also investigate the content of this vesicle at the surface. And we recently developed a cocktail of candidates which permit to isolate specifically 3% of the vesicle in the plasma, and we validate the fact that this vesicle comes from the brain and contain material from the brain. So our ongoing project in Lausanne it's a beginning of the story, but it's a long story to explain. But now we try to investigate with this vesicle different questions about the conversion of the dementia, the fact that you stay in a minor phase of the dementia during very long time, or that you change in a major dementia. But we have also different questions with the Professor Van Gutten about the anxiety and the depression during this process of dementia. But what is the future? And just to finish, because we talk about innovation, what is the future of the dementia and diagnosis? And to explain the future of the diagnosis, I won't talk about and talk about saliva, because as you know, we have different biofluid in our body. Of course, the blood is very known for Alzheimer's disease, but we have also the saliva, and the saliva is the least invasive biofluid, and it's very practical for different reasons. But the thing that we recently analyze the data with metadata analysis, and we observe that the saliva contains approximately 10% of the elevated protein from the brain, exactly the same percentage as the plasma, suggesting that the saliva is also very interesting for the diagnostic of Alzheimer's disease. And we are not alone to think about that, because different publications recently suggest that the phosphoto and the amyloid are also increase or decrease in the saliva of patients, suggesting that we can use the saliva for the diagnosis. And very recently also a PhD in my group and a group of Lavinia demonstrate that the other biomarker like the TTR are also elevated, reduced in a different, with the score of the memory that we can observe in the patients. So just to finish about this presentation, the saliva is perhaps the revolution for the brain monitoring. And we talk about monitoring, but not perhaps diagnosis, because the saliva you can collect at home, you can collect every day, and you can also evaluate more easily the biomarker. So thank you so much for your attention, and thank you for all collaboration in Lausanne and everywhere in the world. And thank you for your attention.
Oh, thank you, Kevin. If I may ask a question directly to you, I was really intrigued by this seeding activity of your brain-derived extracellular vesicle in the in vivo model that you published. Where was the topology of those seeding EV-derived tau following the limbic system? Meaning, was the dissemination of tau similar to what happens in real life?
Yeah, it's a complex answer, because in fact, in this publication, we isolate the EV from different tauopathy, okay, where we know we have different seeding effect of this. So, because if you consider the different tauopathy, we have a clear seeding effect in Alzheimer's disease, where the progression is very clear. The progression is very clear because it's approximately always the same pattern. But it's not the case for the other tauopathy. For example, the PSP, where we have a lot of heterogeneity of the progression of this pathology, and it's more anarchic progression in PSP. That we demonstrate in this paper, that is, in fact, the seeding effect of this vesicle is only observed in Alzheimer's disease. So this different form of tau that we have in this vesicle, we know we have tau in all vesicles in all sorts of patients, but the seeding impact of the tau is only when you choose vesicle from late stage of Alzheimer's disease, meaning that perhaps at a certain form of Alzheimer or stage of Alzheimer, you have some form of tau who can induce a seeding, but it's not the case for the other tauopathy. And you know, different publications suggest that also we talk about tau protein for different tauopathy, but we know that this tau protein is not the same. The conformation of the tau aggregate is completely different if we talk about FTD compared to Alzheimer. So it's perhaps the reason why we are confused about the fact that if we try to diagnose the tauopathy with a biomarker developed for Alzheimer, it's always difficult to see something, because we develop biomarker, PET tau, etc. for Alzheimer's disease mainly. If you try to use for the frontotemporal dementia, it's very complex to have a clear pattern. Or yeah, so it's so I think the question is complex, but yeah.
Okay, so if you were able to extract the brain-derived EV from blood, and then they would have in the cellular, in the dish seeding activity, you would be able to say this is
it's a very good question, and we have also this idea to test, but the big question is, could you isolate enough vesicle in the plasma for this sort of experiments? Because for seeding assay, the quantity of material that we use from the human brain is huge compared to the quantity. So perhaps if we have a liter and liter of plasma or saliva, we can imagine this assay, this seeding assay. But yeah, I don't know if we can have enough material for that.
Sure. Okay, thank you, Kevin. It's wonderful.